All right. Good afternoon and welcome to the Bioengineering Distinguished Seminar Series. Really, it's my pleasure and privilege to introduce Dr. Alyssa Panitch, Department Head of Bioengineering at Georgia Tech. Dr. Panitch has a bachelor's in biochemistry and a bachelor's in polymer science and engineering. And a PhD also in polymer science and engineering space. Then she did her she did her postdoc in Switzerland, right? And and then started off, So I'm sure that was a very exciting experience. And then she started off as a faculty in Arizona. Then trickled down to Purdue, and I was there as a faculty, and we were colleagues for about 10 to 12 years. So what I can say is, she's, as you can see, she's a great leader. She's also an excellent mentor, and so that's been very, very valuable, for a lot of the students she supervised, and I did, had the pleasure of hiring one of the students you supervised, Alyssa, back in Purdue, right here. And so right now, she's here with us, and she's going to share her experiences in tissue engineering, and how do you bring polymers and tissue engineering and such. So it really is a pleasure, and we welcome you to UIUC via Zoom, and hopefully next time in person. And take it away.
Wonderful. Well, thank you very much for the kind invitation and for the introduction. I'm really sorry that I am not there in person. As I mentioned, we have a large workshop going on here, and there was just the conflict was one that was a little bit challenging for me. So I appreciate the opportunity to visit via Zoom. I'm trying to turn on my laser pointer. Hold on one sec. Okay. Hopefully I can point on my little slides and it shows up on the big slide. It does. Okay, very good. My camera's here on my laptop, and there's a big screen over on the other side of the office. So I apologize if I'm looking in different directions periodically. But again, thank you for the invitation and for the opportunity to present some of the work that my group has done over the past several years. And I will look forward to coming to visit everyone in person sometime in the future. Since about 2006, the group has really been focused on developing biopolymers based on glycosaminoglycan backbone. So at the time, there weren't a lot of people studying the glycosaminoglycans. They're really complex molecules, and perhaps the next level of complexity in biology when we think about RNA and DNA, which we understand the sequence of quite well, with maybe some variations with, oh my goodness, methylations and other things that happen to DNA. But we understand the sequence, right? There are 4 nucleotides that make up DNA, 4 that make up RNA. Proteins are made of 20 naturally occurring amino acids. We know the precise sequence that make up proteins. The glycans have multiple different sugar monomers that are sulfated in different locations, and we don't understand the code that biology uses to put those molecules together. So in part for that reason, people hadn't studied them very closely until much more recently. But we realized that the proteoglycans were a very important piece of the extracellular matrix, and the glycosaminoglycan chains themselves, the sugar chains, aren't typically found in the extracellular space just as sugar chains. They're attached to a protein. Hyaluronic acid is the one exception. So we set about trying to take glycosaminoglycans and modify them with short peptide sequences to try to reconfirm the function of some of the larger proteoglycans, which are very difficult to synthesize at scale. So they're made in mammalian cells, but they're not made the same way in, say, insect cells, and they're certainly not made in E. coli and yeast. So making them at scale is challenging. So thus the chemical synthetic approach. We've made several of these molecules. We started off with one called Decarin that I'll come back to a little bit later in the talk. And it's a very simple molecule that controls collagen organization and assembly in the skin and the vasculature and in other tissues. aggrecan, which is very important in cartilage for compressive strength and to protect cartilage from proteolytic breakdown, lubricin, which serves to lubricate cartilage surfaces. And then I'm going to talk to you about what I'm going to call right now ECCL, but you'll also hear me call DSIKL, and I'll explain that as we go along. And that's a glycocalyx mimetic. So let's just start very briefly talking about inflammation as a driver of chronic and acute disease. Now we know that inflammation, if we even look at COVID-19, is a huge problem. The body can have a very exuberant response to the viral infection that causes a systemic inflammation. And when that systemic inflammation happens, the blood vessels become very reactive, and that can cause the coagulopathies or the blockage of blood vessels that we're seeing with COVID-19, and can cause some of the things like the tachycardia that was seen in the heart, as well as other disorders. We also see that inflammation can cause downstream fibrosis. So we see that in cardiovascular disease, and I'm going to focus in that area. But inflammation plays a role in many different disease types. So if we look at what happens in the vasculature during inflammation, which is the area that we focus on, and we look first at the cartoon, we can see that there's a monolayer of endothelial cells within the vasculature, and the endothelial cells are coated by a sugar coating that's called the glycocalyx. And that has that glycosaminoglycan that I was talking about that's conjugated to a protein core and sits on the surface of the endothelium. And if we look at a transmission electron micrograph of the inside of a blood vessel, this is the endothelial cell lining, and these hair-like structures are the glycocalyx. They come off the endothelium into the lumen of the blood vessel. And this glycocalyx serves to essentially mask the receptors on the endothelial cell surface, which allows neutrophils and monocytes and platelets to roll past the healthy endothelium and to just flow through the vasculature. But when there's inflammation, that glycocalyx is damaged. So it's chewed away, and you don't see the beautiful hair-like structure that we saw in the previous TEM image. And if we look at the cartoon, you see that that glycocalyx is lost, and that allows cells like neutrophils to interact with the endothelial cells and to migrate into the underlying tissue. And if we blow up that cartoon, this endothelium now that's missing its glycocalyx and becomes either dysfunctional or inflamed, you see exposed on the surface receptors like E and P selectin, which I'll be focusing on. There are also other receptors that are exposed like ICAM and VCAM that are also important for some of the processes that I'll talk about, but we'll focus on, as I said, the E and P selectin. So neutrophils and monocytes and platelets can interact with E and P selectin and bind to the endothelial surface. And if there's enough receptor on the surface, and the VCAMs and ICAMs are expressed as well, these inflammatory cells will migrate into the underlying tissue. And if there's an exuberant inflammatory response, you can get fibrosis in that underlying tissue. So that can be thickening of a blood vessel or the kind of scar formation that we see on the skin. We also see that platelets bind, and when the platelets bind and become activated, then we can get coagulation inside the blood vessel, which is not ideal. These soft clots can break off and go downstream, and you can get stroke or other issues within the body. So this chronic inflammation or even a high level of acute inflammation can be very damaging. If we look more closely at what the glycocalyx is composed of, I mentioned that there are core proteins that sit within the cell membrane. And then there's these glycosaminoglycans that are grafted to the core protein. And again, that's the hair-like structure that we see. And the glycosaminoglycans can be heparin sulfate, they can be dermatan sulfate, chondroitin sulfate. We've chosen to work with dermatan sulfate. The reason being that dermatan sulfate actually looks a lot like heparin, which many of you may have heard of. Heparin is often used during surgery to prevent the blood from clotting during surgery. It can also be used after surgery for people who have had metal stents or plastic EPTFE grafts put in, or if you've had a total hip or a total knee put in, physicians will put patients on anticoagulants for some period of time, which inhibits blood coagulation. And because we're interested in treating this inflammatory process on the surface of blood vessels, we wanted something that was somewhat anticoagulative. So, as I mentioned, we chose to work with dermatan sulfate. Okay, so what happens in the body? If we have a healthy endothelium, we have this nice glycocalyx, and as I had mentioned before, the blood cells will just roll on by the healthy glycocalyx. If the endothelium is inflamed, we've lost the glycocalyx, and these receptors are expressed. And so what we've tried to do is take the glycocalyx, dermatan sulfate, and we graft to that peptides that bind to these receptors and will stick to the inflamed endothelium and look a little bit like healthy endothelium. So that when cells are flowing through the blood, they look a lot like the healthy environment. So unlike the inflammatory environment, we see very few cells that will stick to this inflamed environment because we've essentially put a molecular Band-Aid on the blood vessel. Okay, so I mentioned that I would call this two different things. For the very first part of the talk, I'm going to call this EC seal. We named it EC seal because we looked at it like a deck sealant for the endothelium. And then a second student came in and did some optimization, and you'll see why this was done as I go through the talk. So the first stab at creating a coating for the endothelium or replacement for the glycocalyx was done by James Wadika, who was an MD PhD student who did his PhD work in my lab. And what James did is he worked with dermatan sulfate, and in glycosaminoglycans, there are often carbon atoms that have hydroxyl groups attached to them. And when you have two adjacent carbon atoms with hydroxyl groups, you can oxidize the carbon bond, and it opens up to form two aldehyde groups. And then you can use those aldehyde groups as handles to conjugate. What we used was a heterobifunctional crosslinker. It essentially allowed us to attach a molecule to the aldehyde group and then use a maleimide group to add a peptide. So fairly complicated, but very controllable chemistry. And he used a peptide that was found in the literature that was developed by phage display. So it's all composed of naturally occurring amino acids. And I forgot it was animated. And so I'm going to skip forward to after he had done a whole lot of work optimizing the molecule and showing it bound to selectin receptors, to show you some work that he did in collaboration with Scott Simon's lab at the University of California, Davis, in collaboration with V. Maricas, who is now a postdoc at WashU in Seattle. So they used a microfluidic device, and they put endothelial cells down on the surface of the device. They then inflamed the endothelium and flowed human neutrophils over the surface of the device. And what they found was if the endothelium is inflamed and they flow neutrophils across the surface of the endothelium, they see several neutrophils that will stop on the endothelium and some that will try to migrate into the surface of the endothelium. If the inflamed endothelium is first treated with the EC seal molecule, we reduce the number of neutrophils that roll along the surface and reduce the number that try to migrate into the surface. And if we use a small molecule pan-selectin inhibitor, it works similarly. So with some data suggesting that it worked in vitro to inhibit neutrophil binding, he worked with Craig Gorgin's lab at Purdue University in a deep vein thrombosis model. Now, the deep vein thrombosis model was really important to us because it was a model that could show us that we were inhibiting clotting within blood vessels in a very rapid way. I don't think that this molecule is ideal for treating deep vein thrombosis. But this model was developed in Tim Wakefield's lab at the University of Michigan and is found to have very similar pathologies to human deep vein thrombosis. So if you ligate the inferior vena cava of a mouse about 90%, you see a ballooning out of the blood vessel And where the ballooning occurs, you get disrupted blood flow. And that disrupted blood flow activates the endothelium, so it becomes inflamed, and you get expression of the VCAM and ICAM on the surface, as well as the selectin receptors. So we thought that this model would be a good model to see if our molecule was working. So we created the ligation in 6-week-old mice. After about 10 minutes, we injected the EC-seal molecule via tail vein and hoped that the molecule would go to where it was needed, where we'd created the site of thrombus. And you can measure the rate of thrombus formation and the degree of thrombus formation using ultrasound. So Craig Gordon's group did this for us. And if we create the ligation and just let thrombus form for 6 hours, you see a fairly large thrombus formation. And if you first treat with our EC-seal molecule, you see a reduced amount of thrombus formation. Similarly, if you fully anticoagulate the animal with heparin, you see the same reduction in thrombus formation at 6 hours. However, at 24 hours, we still see a reduction in thrombus formation using heparin, but our EC-seal molecule doesn't work as well. And this is just an image of the thrombus that we took after sacrificing the animal. And to show that the concentrations of the EC-seal that James was using didn't systemically coagulate the animal, suggesting the molecule worked locally, he looked at clotting time of animal blood. And if we dose the animal blood with heparin, you dramatically increase clotting time because heparin inhibits clot formation. But if you dose with increasing concentrations of the EC-seal molecule, he did not see much inhibition of clotting at all. And we were working down here with concentrations in vivo. So it suggests the molecule is working locally. But as I mentioned, there's some limitations, right? At 6 hours, it seemed to be working really well, but at 24 hours, it wasn't working so well. So there are two potential reasons for that. The first is that using the chemistry James was using, he opens up the glycosaminoglycan, and this can cause the GAG chain to be unstable and susceptible to hydrolytic degradation. And he was conjugating a peptide that was made from all L amino acids, and peptides are susceptible to proteolytic degradation. And most certainly in inflamed environments, there are enzymes that can degrade the peptide. And so we wanted to see if by trying to develop a peptide that had both D amino acids and L amino acids, we could change the chirality of the peptide to inhibit some of the enzymatic degradation. And if we could change the conjugation chemistry to maintain the GAG integrity to reduce degradation of the molecule. So Tima Dagani did this work, and she started off working with Kit Liam's group at the University of California at Davis. And when Kit was a postdoc, he developed a very clever combinatorial peptide approach that was an in vitro approach, so that you can incorporate both natural and unnatural amino acids and isomers of the amino acids, so we could incorporate D amino acids and L amino acids. You can't do this with phage display because the phage only use the naturally occurring L amino acids. Let me talk you through how the library works to give you an idea of what Tima did. So when you make peptides using solid state chemistry, you grow the peptides on individual polymer beads. So every bead grows multiple peptide chains that are all identical. And so if you take multiple beads and you conjugate, say, an alanine to them, each of those beads has many, many peptide chains that are growing that start with an alanine. You can then wash away the resultant alanine, deprotect the the rest of the alanine that hasn't conjugated to the bead, deprotect the alanine, and add another amino acid. And then that conjugates, you wash away the excess, you deprotect, and you grow. So what Tima did is she split the beads into 3 pots. And in the first pot, she added an alanine. In the second, she added a glycine. The third, she added a valine. This is an example. She actually had 30 pots, and she added 30 different amino acids. And then after adding the first amino acid, she mixed all of the resin together and separated it out. And so the resin in pot 1 then had either an A, a G, or a V as its first amino acid, and then she added an alanine or a glycine or a valine, mixed all the resin together again, split it back out. And so you can see here that you're now growing beads, and each bead has an individual distinct peptide chain. So once she did that, and this allows you to make millions and millions of different unique sequences on different peptide beads very quickly. You can then take the individual resin beads and glue them to a dish and probe the beads with your molecule of interest. So in Tima's case, she was interested in peptides that would bind to E-selectin. So she probed with E-selectin that was conjugated to horseradish peroxidase, and then she could add a development agent, and beads that had the E-selectin bound to it also had horseradish peroxidase, and they would turn brown. So you can then take those brown beads and do microsequencing to identify the peptide that was on that bead. In doing this, you can identify sequences that bind very strongly to E-selectin, and then you can pan those different sequences against other molecules to find sequences that are specific to E-selectin. So after doing all of this chemistry, she identified a sequence that's arginine, hydroxyproline, D-proline, leucine, D-lysine, and isoleucine. So this sequence contains 2 D amino acids, so it should be much less susceptible to enzymatic degradation. As I mentioned, she also changed the conjugation chemistry. And so she took dermatin sulfate, and instead of opening up the ring structure of the hydroxyl group, she reacted with this carboxylic acid group. So she used an activating agent called DMTMM. The full name is down here. If anyone else would like to pronounce it, feel free. I'm not trying. This molecule will activate the carboxylic acid group and then allow her to directly add a peptide that has a hydrazide terminus. So she synthesizes all of her peptide with a hydrazide terminus, and we get direct connection of the dermatin sulfate to her peptide with an amide bond, which is the same bond that connects the amino acids within the peptide. So it's a very stable bond. I'm going to point out one other thing about the chemistry that she chose to use. And I mentioned that she used DMTMM. Many researchers use EDC. She chose not to do that because of a very specific concern with EDC chemistry. So EDC can also activate carboxylic acid groups, and it forms an O-acylurea group. However, it can go through an isomerization to an N-acylurea, and when that happens, the EDC becomes permanently conjugated to the glycosaminoglycan. The EDC that stays in O-acylurea conformation can then be replaced with the peptide hydrazide, and you still get your amino or your amide bond here with the peptide, but you get some fraction of the dermatin sulfate carboxylic acid groups that are permanently attached to N-acylurea. This is problematic because it alters the biological function of the glycosaminoglycan. It's going to take you into a completely different project for 2 slides to give you an idea of why this is a problem. So Michael Newen, when he was in my group, worked with chondroitin sulfate and hyaluronic acid. And he was putting thiol groups on chondroitin sulfate and hyaluronic acid because he wanted to form glycosaminoglycan hydrogels for cartilage tissue engineering. And so he was controlling the degree of thiolation. And he would thiolate chondroitin sulfate or hyaluronic acid with about 17 thiol groups, 17% of the carboxylic acids modified to thiols, or about 35% of them modified to thiols. And when he does that and he tries to degrade these GAGs with hyaluronidase, both GAGs being susceptible to hyaluronidase degradation, what he finds is that the GAGs that have a higher degree of modification are less susceptible to degradation because the enzyme doesn't recognize the GAG and can't degrade it. So that's true for both hyaluronic acid and chondroitin sulfate. He then tried to bind peptides that are known to bind to chondroitin sulfate and find that fewer of these chondroitin sulfate binding peptides bind to the highly modified chondroitin sulfate than bind to the highly modified, the less modified chondroitin sulfate, and sees similar results with hyaluronic acid. So for Tima's work, she wanted to preserve the anticoagulant activity of the dermatan sulfate, and that ability to prevent neutrophils and things interacting with the surface of the endothelium. So she wanted to retain as much of that biological activity as possible while adding enough peptide to make sure that the gag bound to the surface. And just one final final piece of information as we move forward. Tima, like James, also looked at the anticoagulant activity of her molecules, and you can see that, again, as you increase the amount of heparin, you anticoagulate blood. As you increase the amount of dermatan sulfate, you can anticoagulate blood. It takes a lot more dermatan sulfate than heparin, but it works. And her modified dermatan sulfate, her now DSIKL, you need even more of it to start to anticoagulate blood. So even just by carefully conjugating peptide to the dermatan sulfate, she is inhibiting some of the biological activity. So again, this is why we want to be very careful about how we modify our glycosaminoglycans. Okay, so let's get back to the project at hand. So remember, her goal is to form this kind of band-aid structure so that we inhibit inflammatory cells from interacting with the endothelium. So now we're going to go to work we did in collaboration with Steve George's lab on microfluidic chips. And Steve is at the University of California at Davis. And they have this very clever chip. It doesn't look very high-tech, but it actually is. They're outer chambers where you can put a gel or you can put a tissue type of interest, and an inner chamber where there are little ports between the chambers that would allow cell migration through those chambers into the outer chamber. And in this inner chamber, we can put a monolayer of endothelial cells shown in red, and then we can flow neutrophils through this chamber to look at how many neutrophils interact with the endothelium. And we did a lot of work to figure out how much TNF-alpha and IL-1 beta we needed to add to inflame this endothelium so that neutrophils would interact with it. So we could test whether our ECCL molecule, not our ECCL, our DSIKL molecule that Tima designed, would inhibit neutrophil interaction with the endothelium and then migration into the underlying chambers. So the first data I'll show you, this is just filled with a gel-like solution. And the concentrations of TNF-alpha and IL-1 beta that we ended up with are similar to what you see in patients with severe COVID-19. Okay. So if we look at these chips using confocal microscopy, if we just inflame the endothelium and flow human neutrophils across the endothelium, you see lots of neutrophils interacting with the surface of the endothelium, and you can see neutrophils that have now diapediced across the endothelium out into the outer chambers. And as we treat with increasing amounts of our DSIKL molecule, we start to inhibit neutrophil interaction with the endothelium and diapedesis out into the outer chamber. And just to show you what that looks like, let's see if I can get these movies to play. No, I don't want my laser pointer. Let's see, can I get rid of it over here? 1 second, I'm sorry. Nope. Oh, there we go. Okay. So if we treated with our DSIKL molecule, you can see neutrophils flying across the surface. And if we don't treat with our DSIKL molecule, you see these neutrophils are crawling very slowly across the surface of the endothelium. And you can quantify rolling speeds and see that the neutrophils roll very quickly if we have the DSIKL molecule present. And then again, this happens in a dose-dependent manner. Okay, let's see now if I can get it to stop. Okay, so the next thing Steve's lab did is they took fibroblasts and created iPSC cells, so induced pluripotent human cells, and then differentiated these cells into cardiomyoblasts. And those are cultured in the outside to mimic heart tissue. And they're co-cultured with cardiac fibroblasts to help maintain the differentiation of the cardiomyoblasts. And then we did the same experiment. We inflamed the endothelium and we flowed through neutrophils because we were trying to start to understand how neutrophils may create arrhythmias in patients with COVID-19. I'm going to just show you one piece of data. And that is, if you take our chip and you do not treat with an inflammatory agent or with DSIKL or with neutrophils, you see a standard beat rate. If you inflame the chip, you don't see any change in that beat rate. If you inflame the chip and you treat with DSIKL, you don't see a change in beat rate. If you inflame the chip, don't treat with DSIKL, but treat with neutrophils, you see a rapid increase in beat rate. But if you treat with DSIKL before treating with the neutrophils, that beat rate does not increase. And what we saw is that we got an increase in the beat rate, but we did not get an increase in the conduction velocity. So the cardiomyocytes were healthy, the conduction velocity was the same, but they were beating much more rapidly. Okay, so that's all the chip data I'm going to show you. I'm now going to move into an in vivo model, which was the model we were interested in back when James created the first molecule. And this is ischemia reperfusion in coronary tissue. So when a person has a blockage in the coronary artery that leads to a heart attack, that blockage causes some degree of cardiac tissue death. And what physicians do is they go in with a balloon and they compress the plaque that's formed in the coronary artery and reestablish blood flow. Well, when they reestablish blood flow, there's something that's called a reperfusion injury that happens, and you actually get a larger area of damage in that coronary tissue. And that happens because, in part, because the endothelium becomes very inflamed and you get lots of inflammatory cell interaction with those coronary arteries. And so we worked with Nip Chiambimavat's lab at UC Davis, and she has a very talented microsurgeon, Feng Tai, who works in her lab, who created a model for us where he ligated the left anterior descending artery for 40 minutes in a mouse model, and then released the ligation and allowed reperfusion, and then closed up the animal, and then injected our DSIKL molecule through the tail vein about 10 to 20 minutes after the ligation was released, and then again at 24 hours. And we followed these animals at 24 hours, 36 hours, and at 2 weeks. And we also have a sham control where they open up the animal, they don't ligate, and they close the animal. And then we have animals that are just treated with saline and not with the DSIKL molecule. And so we're looking here at an in vivo imaging system where we have labeled our DSIKL molecule with a red chromophore. And so we can start to see where the molecule is going in the body. And after 24 hours, so 24 hours following injection, we still see some systemic distribution of the molecule, but we see accumulation of the molecule in the heart. And we don't see any of that same fluorescence in animals that are treated with saline. Oops, sorry, I went the wrong way. And we can quantify this based on background. And so we see our sham control animals that were treated with DSIKL. And this is quantification at the cardiac tissue. If we have our ligated animals that were treated with saline, we don't see accumulation of fluorophore, not surprisingly. But if we treated with DSIKL at tail vein, we do see accumulation at 1 hour, at 12 hours, and at 24 hours. So the molecule is staying at 24 hours. Now, you know, hindsight is 2020. If we had analyzed the data at 24 hours and could have made a quick call, we would not have dosed the animals again. But our protocol was to dose again at 24 hours. So we dosed again at 24 hours. So at 36 hours, we still see the molecule there. But we know we have a stable molecule at least for 24 hours, which was not true in the thrombosis model that I showed you earlier. So Tima's molecule is more stable in vivo, but we don't know for how long it will be stable because we redosed. Okay, so we also looked at the degree of tissue damage using TTC staining, and where you see white tissue, like you see here, that's where the tissue has been infarcted, you don't have blood flow, so that's tissue damage, and that typically leads to fibrosis after a week or two. And we see in animals treated with saline that there's a high degree of or there's more white staining or a lack of blood flow to the tissue as compared to hearts that were treated with the DSIKL molecule, and that is shown here. And we also looked at a blood marker of cardiac damage. It's I-troponin in the mouse, and we see a reduction of that I-troponin in the mouse model when we have treatment. We also took the hearts and dissociated the tissue to isolate the individual cell types. There's data on the fibroblasts and on other cell types as well in this publication. I'm just going to show you the neutrophil data. But in our sham animals, there's a very low accumulation of neutrophils in the damaged tissue. In the sham animals treated with DSIKL, again, low levels of neutrophils, and this is flow data. In animals that had an infarct and were not treated with the DSIKL, there's an increase in neutrophils, and that is decreased when we treat with DSIKL. And this data is again quantified on the right. This is, and again, that was at 36 hours. That corroborates the data that we see at 2 weeks, where if we look at the neutrophil data on the right panel where we see the green and red staining, in sham animals we see no accumulation of neutrophils, but in animals with an infarct that were treated with saline, you can see red neutrophils, and this is an overlap of just a general cell actin staining. So you can see where the cells are and where the neutrophils are, where the coronary cells are and where the neutrophils are. And if we treat with DSIKL, you see very few neutrophils present. And that is quantified here on the right. And we see similar activity with macrophages, where we see an accumulation of macrophages in the infarct tissue at 2 weeks, and we don't see those macrophages in the treated tissue. And again, quantified here on the right. This data agrees with functional data. So we measured cardiac function by looking at fractional shortening. And this is the ability of the heart to contract. So the greater the shortening, the better. And so in sham animals, you see normal shortening, which is just above 50%. In the untreated animals that have an infarct, you see considerable decrease in that shortening. And in the treated animals, you recover some of that shortening. You also see a decrease or a preservation of ventricle function, as shown here. And we see an increase, and this is the ability of the ventricle to relax after it's contracted. So it's able to relax better with treatment. And this is a measure of diastolic function, or the ability of the ventricle chamber to fill with blood so the heart can pump blood back out to the body. And with treatment, we see better filling of the ventricle with blood. So overall, we see positive results with the DSIKL molecule. Again, I want to remind you that we have not anticoagulated the animal with our molecule. So it suggests that the molecule is working locally, where we also saw the molecule accumulating at the heart with the in vivo ultrasound. Okay, so in conclusion, we've been able to design a molecule that's much more stable after we went through iterations to incorporate D-amino acids and change our chemistry. And the molecule seems to behave a lot like the native Glycocalyx to reduce platelet interaction with the endothelium and that soft clot formation, as determined with the deep vein thrombosis model. We're also able to inhibit neutrophil and monocyte interaction with the endothelium, as indicated with the cardiac fibrosis model. And this is the group that worked on this project. And so I think that I have maybe 10 minutes left. If that is true, I'll go ahead and tell you a second story. Okay, I'll try to walk through this story a lot quicker. So in this case, we're really trying to look at wound healing, and this is dermal wound healing. And we're particularly interested in ischemic foot ulcers that happen particularly in patients with diabetes. And these patients, often these ischemic wounds won't heal, and it leads to limb amputation. And it is the #1 cause of non-traumatic lower limb amputation. And unfortunately, there's a high degree of mortality for patients whose wounds result in this lower limb amputation. Now, in these ischemic wounds, there's disrupted angiogenesis, or new blood vessel growth. And there is a whole lot of oxidative damage. There's endothelial dysfunction and fibrosis. So I am going to focus on our ability to induce new blood vessel formation rather than on all of the oxidative stresses. So those are things that we would want to be able to deal with as well. So I mentioned earlier that the first proteoglycan mimetic we made was a decorin mimetic. Decorin is really important in skin function and in skin healing. And decorin has a core protein, and it attaches to collagen fibrils, and it has a gag chain coming off the collagen, or the collagen binding core protein. And the core protein binds to collagen, and it helps control collagen fibril diameter, and the gag chain helps with collagen fibril organization. So we asked the question, could we take a dermatin sulfate backbone, conjugate collagen binding peptides, and start to control collagen organization? And so this TEM is from work in Thomas Iozzo's lab Renato Iozzo's lab at Thomas Jefferson University, where he has wild type mice and mice where decorin is knocked out. And these are TEM images of the skin. And we're looking axially down collagen fibrils. And you can see in normal skin, the collagen fibrils are all about the same size and very uniformly packed. But when decorin is absent, you have multiple different collagen fibril sizes, and they're not packed very well. Interestingly, human fetuses and healthy elderly adults have very high concentrations of decorin in the skin, and the rest of you have lower concentrations of decorin in the skin. And fetuses and healthy elderly adults don't scar very much, whereas everyone else scars fairly readily in the skin. And so there seems to be a correlation between decorin and scarring and collagen organization. Okay, so we did a whole lot of work to show that we could make these decorin mimetics, and in vitro we can control collagen organization. And we went to an in vivo model. So this is a rat model where we put a 4-centimeter wound in the back of the rat. We then drip in our decorin treatment in a hyaluronic acid solution. We use hyaluronic acid here for 2 reasons. 1, because it's viscous and it would hold our decorin mimetic in the wound bed. And 2, because hyaluronic acid is known to be favorable for wound healing. So then we just suture up the wound and look at the wound bed after 21 days. And if we put an incision in and we don't treat with anything and we just sew up the animal, you see dysregulated collagen fibrils by TEM, and we quantified the size distribution of those fibrils. If we just treat with hyaluronic acid, well, the size distribution is very broad and the packing isn't very good. If we treat with our diacili with the hyaluronic acid, we see very uniform packing and a better size distribution. And this is what the normal rat skin looks like. So we're starting to approach normal. If you look at the surface of the wound, you can actually see the scarring and even where the sutures were in the untreated animals. In animals treated with diacili, you see, or a decorin mimetic, you see essentially no scar on the surface, and you actually see very little scar tissue with histology as well. This is outside the scar area. This is inside the scar area. They don't look too different. There's a little bit of dense collagen here, but in the scarred animal, you see a lot of dense collagen right here. So there's correlation with what you see on the surface and what you see inside the skin. And the other thing that I want to tell you is that decorin protects collagen from degradation from proteases. So you can take collagen fibrils in vitro and treat them with a protease. And you see, this is STS page where you can look at the size of collagen fibrils. You see, this is normal collagen fibrils, or normal collagen here. If we treat with a protease, you start to see smaller molecular weight fragments. If we protect the collagen with our diacili molecule, you don't see those small fragments. So we can protect from proteolytic degradation. Okay, so my colleagues at UC Davis have developed this cyclic peptide that binds specifically to the alpha V beta 3 integrin receptor. And they developed this because they wanted to pull out endothelial cells and endothelial progenitor cells from flowing blood to have them bind to the surface of vascular grafts. And only endothelial and endothelial progenitor cells within the cells in the blood express this alpha V beta 3 integrin receptor, so they knew they could selectively pull out these integrins. When I saw this peptide and knew that it bound specifically to this integrin, I was very excited, because we know from the literature that this integrin interacts with a receptor called VEGFR2, and it potentiates this receptor. So what that means is that in the presence of vascular endothelial cell growth factor, which stimulates new blood vessel growth, if you have the alpha V beta 3 integrin receptor engaged, you need very little VEGF to activate this receptor. So you need low quantities of VEGF to stimulate angiogenesis. So what we did was we took our Decarin mimetic and added this peptide to it, so that we could take a molecule that would bind to a collagen scaffold and could bind to endothelial cells and stimulate those endothelial cells in the presence of very low levels of VEGF to induce new blood vessel growth. So we wanted to use this for wound healing. So this is the work of Tanaya Willambi. She still used the EDC chemistry. You've seen it before. She used it to attach both the collagen binding sequence and the integrin binding sequence. And just to give you her nomenclature, DS6 is our original Decarin mimetic molecule. It has dermatan sulfate with 6 collagen binding peptides. 2L-DS6 has 2 of the LXW7 peptides that bind to the integrin. 4L has 4 of those peptides. 6L has 6 of those peptides. So 6 integrin binding peptides, dermatan sulfate, and 6 collagen binding peptides. So if we take endothelial cells and we expose them to collagen and we probe for VEGFR2 activation, we see none. If we take the same cells and we treat them with DS-Cili or our Decarin mimetic, we still don't see VEGFR2 activation. But if that DS-Cili now has 2, 4, or 6 of the LXW7 peptides for integrin engagement, we start to see activation of the VEGFR2 receptor. Consistent with that, we can do a tubulogenesis assay where you take endothelial cells and seed them on either collagen or matrigel scaffolds, and the endothelial cells would prefer to interact with each other than the scaffold. And so they'll start to form these little tubules that are sort of mimics of tube formation that you would see in vivo. And if we have the 6L DS6, we see a greater number of these kinds of loops forming as compared to any of the other treatments. And we see that the cellular area is composed of more complete loops than other cell types. And then if we look at angiogenesis in what's called a chick chorioallantoic membrane model, where we take fertilized chicken eggs and we stimulate angiogenesis on the surface of the yolk, we can put collagen gels or collagen gels containing variants of our LXW7 DS-Cili molecule and look at the degree of angiogenesis. What we find is an increase in new blood vessel formation if we use the 6L DS-Cili as compared to any of the other variants. And we just have the control here of just DS-Cili. So it seems to induce angiogenesis in this particular in vivo model. So we took the molecule and we used an extraordinarily aggressive ischemic wound model. So we took the Zucker diabetic fatty rat, which has a metabolic syndrome very similar to human diabetes. And we created ischemic wounds that were 6 millimeters in diameter on the back of the animals. And there's a silastic sheet underneath the skin flap so that no blood vessels can grow from underneath. They can only grow from the tail or the head. So these are very ischemic wounds. And we have non-ischemic wounds outside of the skin flap area. And then we took Cook Biotech small intestinal submucosa, which is a collagen-based scaffold. We treated it with either the LXW7 DS-Cili that has 6 LXW7 and 6 Cili peptides, or just our original Decarin mimetic, so one or the other. And then we used endothelial progenitor cells or not. And we treated these different wounds. So what are the results? And I have like 2 or 3 slides left. So if we just visually look at the ischemic wounds, if we have the SIS plus endothelial progenitor cells at day 1 and day 15, or the SIS plus the LXW7 DS-Cili molecule plus endothelial progenitor cells, you see much improved wound healing. I'm going to quantify that for you here. And these are a little bit complicated. So first we're going to look at the non-ischemic wounds. These are the top two graphs, with or without endothelial progenitor cells. So with endothelial progenitor cells are the, I don't have my laser pointer anymore, do I? Are this graph. This has endothelial progenitor cells. So non-ischemic plus endothelial progenitor cells, non-ischemic without endothelial progenitor cells. And in both cases, what we see is if you have the SIS with the LXW7 diacilly with endothelial progenitor cells, you get slightly better wound closure at 14 days. So with or without the endothelial progenitor cells. So it seems that if you don't have ischemia, you don't need endothelial progenitor cells, but the LXW7 is beneficial for wound healing. However, if you have ischemic wounds, if you have the endothelial progenitor cells, you see dramatically improved wound healing as compared to without endothelial progenitor cells. And so I'm looking at a really small screen here. So we see about, 40% closure rate without the endothelial progenitor cells, but closer to a 60% closure rate with the endothelial progenitor cells. With the non-ischemic wounds, it's closer to an 80% closure rate. So we're not back to the non-ischemic wound, but certainly a much better closure rate with endothelial cells with our LXW7 molecule as compared to with just the diacilly. So it seems that by engaging the integrin, we're able to induce better angiogenesis. And we have this publication here where all of the data is presented with histology of the wound bed and showing the increased angiogenesis and the improved closure rate. But I just wanted to show you this piece of data here. I'm going to actually stop here and not tell you a lot about where we're going, but where we're going is that we're fairly convinced, because the endothelial progenitor cells only live for about 24 hours in the wound bed, because the wound bed is fairly dry and a very hostile environment. We're fairly convinced that it's the secretome that is responsible for the increased angiogenesis. And more than that, we're convinced that it's actually the extracellular vesicle secretome. And we've been able to show that if we take the secretome from endothelial progenitor cells that are treated with LXW7 and then treat naive endothelial progenitor cells, we do, at least in vitro, increase angiogenesis. And that's just quantified in this data here. So, in a nutshell, that's where we're going with this project. But I'm gonna just stop there and say, we've been able to combine our Decker mimetic with very specific integrin engagement to enhance angiogenesis and wound closure in these ischemic wounds, and really hoping that we can help some of these patients that have these ischemic wounds that won't close on their own. So I want to thank lots of funding agencies, lots of people who did this work. I tried to cite everyone as I was going through the work. And I'm just going to close with a little bit about our department. For any graduate students and postdocs who might be out there, for the graduate students who are looking for postdocs, please come look at our website. We have lots of faculty members doing lots of great projects that are always looking for postdocs. And for the graduate students that are interested in graduates, and for the postdocs who are interested in faculty positions, and even grad students who might be interested in going directly to faculty positions, we're always hiring. So with that, thank you very much, and I will be happy to take any questions if there are any.
Excellent. Thank you so much, and we're on time. We still have about 10 minutes for questions. The floor is open to questions. Feel free to jump in.
I typically find that I've either terrorized everyone in the room with the amount of chemistry and biology and nobody will ask anything, or there are a whole bunch of chemists and biologists in the room who dig into the chemistry and the biology.
All right. So let me start off. Oh, go ahead, Hua. I think.
Hi, Joseph. Hi, Mr. Great talk. Really enjoyed it. So I do have a question on the I know you showed some data on the stability, how long they can the molecule bind to the endothelial layer, how long they can stay. I'm still curious, what's the longest time they may persist? No, either a few days, a few weeks.
Yeah. So, my gut tells me it's probably only there for maybe 48 hours or so, maybe a little bit longer, because there is turnover, right, at the endothelial cell surface. And there's a lot of data in the literature suggesting that if something is at the blood vessel surface for 24 to 48 hours, that's sufficient to allow for normal healing. So my guess is because of turnover, that's probably the time frame. But we have to go back and do the IVUS data again and then extract the molecule to make sure it's intact, which we have not done yet to verify the data.
Yeah, great. Yeah, thank you.
Thank you.
I have a question. In the wound healing, project you showed us, some of the wounds are ischemic. Do you think, in addition to the materials, if we supplement that with oxygen, will the wound healing be enhanced, or is oxygen needed at all? Or
yeah, interesting question. So there is evidence out there, right, that if you can enhance the oxygenation of the tissue as well, you enhance healing. So I imagine that would further enhance healing. Our treatment is not it can be used with hyperbaric oxygen chambers and things that physicians use to promote wound healing. So I think that the way it's designed, it would be consistent with current treatment, but certainly there are different oxygen carrying agents that could be used too in these animal models to see if it would further enhance healing. Yeah.
Yep. We can probably talk later, or some of the
that would be great
in detail on that. Yeah. So we have some projects ongoing. I think. Yeah.
Okay. Yeah, I would love to talk about it.
Okay. Sure. All right, students, come on. Are you still there?
Yeah.
I'm taking attendance. I'm watching you. Please do. I need a few questions here, okay? I want you all to unmute, show up, and we should have the video on too. Nobody. My goodness. How about that?
Well, in that case, maybe I can ask another question. I'm really curious. So related to my first question, so what if we administer your molecule orally? What will happen? So will they bind to the mucin layer? Is it possible to develop a long-term depot vaccine? Potential application for that? I'm just curious. Yeah. What will happen if we take it orally?
Right. We haven't tried to do that just because proteins are degraded so well in the stomach, right, with the stomach acid, that I suspect that our peptides will degrade off the dermatin sulfate. And so we wouldn't get good delivery. The way the peptides are designed, we've really only looked at either, sort of the topical delivery, the dermal delivery that I showed, or IV delivery, because we're concerned about degradation within the digestive tract. I think we need a clever drug delivery person to protect it in the digestive tract so it could be delivered that way.
Yeah, that's true. Yep.
Yeah, thank you.
There is a question in the chat. If you could.
Let's see. I don't see the chat. Do you see the chat?
Okay. Yes,
can you read
it? Yeah, this is from Anna, one of the grad students. So for the mouse injection, after release of ligation, why was there another injection 24 hours later?
Yeah. So, at the time, we weren't sure what the half-life would be, right? We had seen that in the deep vein thrombosis model, the molecule wasn't working as well 24 hours later. So we decided, these are very challenging experiments. The coronary artery on the mouse is really, really small. And so it's just a very difficult surgery. So to enhance our chances of success, we dosed a second time. As I said, hindsight is 2020. So actually we did all of the 2-week experiments before we went back and did the in vivo imaging experiment. And so we didn't know, we should have done it in the other direction, right? We should have done a few in vivo imaging experiments to see that the molecule was still there at 24 hours, and then we probably wouldn't have dosed a second time. But we were hedging our bets when we did the study.
Great. Thank you. Okay, one more question, a couple more.
Yeah, so now I see them. I'm not sure why I didn't see the first one.
Yeah,
so
okay.
I'll answer the one that came up for me first. And that is, I'd like to ask how you fabricate the microfluidic chip and grow cells inside it. I've been trying to grow a 3D endothelial cell layer, but they start to clump into a group and don't spread out. Yeah, so that work was done in Steve George's lab. And they have a lot of experience with these. And so that's unfortunately a question that I can't answer. But if you would like to email me, I'd be more than happy to put you in touch with folks in the George lab to see if they can help you with that. I'm sure they would be happy to do so. And then there's some folks here too, who do some lab on a chip work with endothelium. So if the George lab can't answer, I can tap one of my colleagues here. So we studied the effects of combining our treatment with vitamin C administration. No, we have not. I showed you our most recent data, so we haven't had an opportunity to start looking at how our molecule might be additive with other treatments or synergistic with other treatments. But it's a very good question and something that we should start looking at.
Great. All right. Any other questions at all? We have maybe just one more question. I know that we are almost on time, but. Mark, were you saying something? Mark was muted, so.
Oh, no. So I thought it was an interesting talk, but this is, outside my wheelhouse. So I don't have any intelligent questions to ask. But very interesting work. That's for sure.
Thank you. It's one of the things with biomedical engineering, right? It's really, really broad. And, we have people who do imaging and people who do more of the biomolecule things like I do, and more of the tissue engineering and diagnostics. And so it's, as I said, I either have an audience that looks at me blankly because it's filled with a whole bunch of people who do very, very different things, or I have an audience that's filled with chemists and biologists who ask me a whole bunch of really, really difficult questions.
Right. Yeah,
I
somehow I feel that people doing chemistry and biology in our department are more shy than the people doing imaging and signal processing, because the imaging speakers get hammered when they come in. I'm not exactly sure why, but yeah.
No, funny.
Great.
But thank you all again so much for the opportunity to present my work, and apologies for not being there, but hopefully we can find a time when I can come visit, and I don't have to give a seminar, but chat and learn more about the department. It would be wonderful.